Okay, all right. So Ali, you sort of preempted me as to the sneak peek that I'm going to give you guys. I'm going to talk just, I guess, for 5 minutes, very briefly about this plasma proteomics project that we've done across many rare neurodevelopmental disorders, including obviously KCNT1 related disorder. And I should say that this is a really cool project that was funded by various industry members and organizations, some of which are probably on this call, and they have graciously allowed us to share this data as well as this data set to many of the other researchers and academics here. So I have to have a big shout out to them making this project possible. So that in mind, and given the small amount of time that I have, I'm gonna skip the background on what biomarkers are and how they're really critical, a critical component of the drug development process, and just skip right into the overall aims for this project. And those can really be sort of bucketed into 2 or 3 different primary aims. And the first one for me and our team was really to identify shared as well as unique biomarkers for rare neurodevelopmental disorders from patient blood samples. And for this particular project, when I mean biomarkers, we're looking specifically at plasma proteins with a particular interest and emphasis on neurospecific proteins, or otherwise proteins that originate from the central nervous system. And then the second aim is really to understand the impact of age and sex and epilepsy on the plasma proteome. And then the third one, which is really sort of related to that, but something that's really pertinent and relevant to KCNT1 and other developmental epileptic encephalopathies, is can we start differentiating the effect of various pathogenic genetic variants from the effect of epilepsy on, in this case, the plasma proteome. Okay, so this is very much a simplified overview of this study design. But our goal was to try and collect 20 blood samples from various different neurodevelopmental disorders and to get at least 40 different samples from aged matched controls. We collected an ADTA tube of blood from all of our potential patients. We processed it into plasma almost immediately. We aliquoted it, and then froze it right away. In addition to collecting blood samples or other biological samples, we also had participants complete a variety of different phenotypic assessments so we could sort of get a gauge on seizure burden, diet, special diets, particularly kids with epilepsy are on, and then everybody's favorite neurodevelopmental assessment, the Vineland Adaptive Behavior Scale. So from the plasma that we collected, we used one small aliquot of that and sent it off for plasma proteomics. We used this SomaScan platform, which the readout for that is the relative expression for about 11,000 different proteins. So this again is a snapshot on how we collected all of these samples. We primarily went to family and patient conferences, which Ali suggested, sort of talked about how they're getting groups of community members together. You can see on the right-hand side here, a way less cool version of the ERAS tour, the dates where we collected these samples. We also did a fair number of home phlebotomy visits where we had someone come to the participants' home. And then we also had a handful of really awesome clinicians who helped us coordinate alongside clinical lab draws, a research blood draw as well. So we collected samples basically for, ultimately almost the better part of a year, closer probably to 10 months. And if you look here, this is kind of a horrendous, it's a big table, but this is the breakdown for all of the participants that were involved in this project. So if you look on the left-hand side here, you can see the outline of the different disorders that participated in this project. hopefully you guys can see my sort of pointer going over this. The number of participants in each group, the average age in years, percent female, and then the number of participants who either reported current or past medical history of epilepsy. So I've highlighted controls as well as the KCNT1 related epilepsy samples. And there were actually 11 samples donated, but one was sort of dropped from the analysis because of assay issues. Approximately 4 years of age, 20% female, and then no surprise, 100% of patients reported current or previous epilepsy. And well over our goal of 40 control samples, but with an average age of about 16